absolutely excited to participate in the Spectrum event. Looking forward to describing our newest effort here at Scripps Research. So this will be a co-presentation between myself and Katie Baca-Motes, and we figured we can start out with a quick introduction that not only speaks a little bit beyond our bios, but also how we kind of entered into this initiative and made it come to life. So for me, I think about my research pathway as a bit of an evolution in almost literal terms, kind of working on the microorganism side of things early in undergraduate, kind of evolving then to gene expression in mice and nuclear organization in human tissue samples. I always joke at a deviation then to work on in the United States Senate and Congress, where I may have had then a misstep in evolution before then going back to research and having an opportunity at the National Institutes of Health to then expand work on the genomics side of things, working with population genomics in terms of cohorts, and then growing that beyond an understanding of not only the population and individual level, but what other data then influences health and disease, and very specifically in terms of leveraging digital health devices and sensors, and then ultimately putting all of that together. And so I'm excited to not only take my research background, but consistent themes that also persisted throughout this amazing journey, which included things like exposure to science policy, ethical, legal, and social implications of the work that we do. I am deeply invested in ensuring that we integrate a lens of health disparities in all our work. And so extremely excited that the Scripps Research Digital Trial Center that Katie and I have launched give us an opportunity to really integrate all of those different experiences and skill sets and bringing them to bear. So I'll kick it over to Katie for an intro, and then she'll start in on the presentation, and then I'll come back to describe a little bit more about the research and science that we're doing at the center. Katie?
All right, thanks, Ed. So my background is a little less conventional, at least as a way to get into clinical research. Back when I was in business school, 2007 to 2009, I sort of fell in love with behavioral science and behavioral economics. And it landed me my first job out of business school, which was a Walt Disney Imagineer, running randomized controlled trials, really trying to understand guest behavior in hotels and in parks. So that was my first sort of hybrid sort of academia industry experience. Loved it there, was there several years. During my time working with a couple of academic partners, I was actually introduced to Dr. Eric Tupel and Dr. Steinhubel, and Dr. Steinhubel was originally the one who hired me to work as a consultant for one of their MSTOP studies. So I don't know if it was just because Steve is a big love of Disney. He and his wife go there all the time, or he's also a fellow admirer of behavioral economics. But he called me up one day and said, hey, we're working on this MSTOPS trial, which is a remote direct-to-participant trial for people over the age of 65. Can you come in and help with some of the recruitment and enrollment and maybe put a behavioral science lens on it? So that was my introduction to the world of clinical research, and really took off from there. Spent the last 4 years working along with Ed also on an NIH program. It's the All of Us Research Program, which is really, like this says, really the largest, most diverse medical research program that's ever been run. So we have about 380,000 participants across the country, 80% of those coming from a background that is traditionally underrepresented in biomedical research. From there, we're taking that now, and we're super excited to talk about, some of our other initiatives. We've taken a lot of learnings from the past research studies that we've done, and really bringing that all together in what we're going to talk about today, which is our new Scripps Research Digital Trial Center. All right, so it was interesting. Ed and I were kind of laughing at this categorization of this New York Times article that came out just a couple of weeks ago, It was filed under the unintended consequences of COVID, and Lord knows there's been a lot. But this idea that clinical trials are moving out of the labs, out of the sites, and into people's homes. But here, like we were already starting the idea of digital remote trials, like I said, with the mSToP study and others much earlier than that, the Welderly study. Here, this has really been a paradigm shift, years in the making, from, you know, Eric's first books on creative destruction of medicine and the patient will see you now. How can we really get direct to participants, direct to patients? How can we change the way that clinical research is done? So here's just a snapshot of a couple of the research papers that have come out from our group over the last several years, looking at, you know, detecting AFib out in the real world. That was our mSToP study, looking at characteristics of the modern day physician house call. That was with a partnership with the company Heal, where we're taking actually like doctors on demand and making sure we're getting back to, you know, what is now the house call, the more modern of that. The All of Us research program, we've done things with tracking pregnancies through digital sensors. We have lots and lots of studies that are looking at how can we capture real world device data using wearables, using smartphones, using new novel sensors. What are the things that we can start capturing on an individual that takes it out of the clinic and really into the home or into the wild or into any other convenient location. Next slide. So about, I would say, I don't know, about a year and a half ago, we started kind of building this portfolio, which is now our portfolio for the Scripps Digital Trials Center. We were partnering with the My Data Helps platform, which is a platform from the company Carevolution on the All of Us research program. What you're seeing here is the direct volunteer app where people can enroll anywhere in the country, enroll straight from their phone or a website, and start to contribute things like their wearable device data, electronic health records. We had our Power Moms study. Most recently, I'm going to talk about in just a second, we have a COVID study called Detect. And really, you know, one of the newer partners that we have, because we have this network of great collaborators, probably some of them are on the call. We have this great network of probably, you know, a couple of dozen, maybe more partners across the country that we partner with on several of our studies. We call them the usual suspects that we go back to time and time again because it's such a great network of collaborators. But our most recent one is really something that's going to accelerate a lot of the work that we do in the digital clinical trials. It is this Carevolution My Data Helps platform. So Vic Ketterpal, this principal there, has come with us, and they actually bring a lot of expertise on the technology side and also on the clinical research side. So they're the back end for the electronic Framingham Heart Study, the MyPAC study, the Rural Cohort Study, and several others. We're really excited about that partnership. And what this does, so this partnership with Carevolution and our other outreach partners, our participant partners, is really laying the foundation for transforming clinical research. And, you know, for those of you who might be listening in and saying, you know, well, a lot of people are doing that. This is true. It's not just about going digital, as we saw in that unintended consequences. There's lots of amazing organizations who are taking clinical trials and turning to remote siteless clinical trials so we can start to get to scale. But really what we're doing is trying to accelerate this in a way that leverages you know, all of our partners outside of the traditional academic medical centers so that we can start to enable an infrastructure, at first here in the US, but eventually globally, to allow anybody anywhere to raise their hand and say, I want to contribute to and benefit from biomedical research. So we do that by a real scalable approach. So we take it out of the sites. You don't need to be near an academic medical center. We partner with things, with companies like Quest, like Walgreens, you know, organizations that offer in-home biosample collection. We actually do patient-generated biosample collection via salivary kits, or even I've even tried the tap devices, those draw your own blood devices on your arm. Those are the new things that we're trying out. Technology is really one of the foundations of Scripps research, looking at the wearable sensor data and the new novel sensors. And then also embracing this whole patient-mediated EHR capture via the Smart on Fire API and allowing that data visualization to come back. And then really the part that bringing it back to sort of my foundation is the participant experience. One of the maybe drawbacks of working early on in a company like Disney, where your guests are literally like tattooing Mickey Mouse on their bodies, and just seeing that engagement with the company, with the brand, with the experience is really powerful to me. And I think it's a lofty goal, but I think we want to take that sort of magic, right, and inject that in something that is so critical for the health of our nation, making that participant experience something that people will be proud to be a part of, feel connected to, get value from back as an individual and also as a community. So again, a lofty goal, but I think it's something that we're definitely trying to do with not only just the behavioral science element, but bringing in novel things like A/B testing and just trying to figure out a way to continue to test, to iterate, to improve all of our experiences. So this is our latest and greatest, which, like probably many of you, you've had to pivot and really focus this last year. I don't know, sometimes I think it's hard to believe it's already been a year since the start of the pandemic, or it's only been a year, depends on the day. But this was our response. And so we started the Detect program, started thinking about it back last February, so just over a year ago. Flashback to the time, we were still in the office, actually meeting face-to-face, talking about these things. And if you go to the next slide, this is sort of how it started. We were all sitting around, we had all of this great idea around the Digital Trials Center. How quickly can we enable patient-centered research very nimbly, very quickly with this new set of partnerships? So this was our use case. We were very excited to see, okay, it's a great idea, but can we do it? So what we did know back in February, March, there was some really nice timing from a couple of papers that came out from Dr. Jennifer Radin, Dr. Giorgio Quer, or one of our partners for the last several years has been Fitbit. So we've been working on a de-identified anonymized data set, looking at things like heart rate, sleep, activity. And Dr. Radin and Dr. Quer have been leading some great work. And just in February, I think it was the last week of February, or first week of February, last week of January, we had two timely papers come out. One looking at a variation in heart rates as an individual, showing that, anywhere from 40 beats to 100 beats per minute could be normal. Your normal heart rate only varies about 3 beats per minute. And for those of you, I might have got those numbers wrong, but somewhere in that threshold. The other one that came out was Jennifer Radin's work. Previously, she was at the CDC, and then she's been at the Scripps Research for a long time, looking at, can you use this population level data, looking at resting heart rate, and see if you can start to predict flu outbreaks. Turns out you can. So those came out perfect timing with the pandemic. We started getting calls from our partners, hey, this is really timely research. Do you think this might be able to help us with the new COVID pandemic? So a couple of late nights, weekends, very quick protocol writing, wonderful IRB process. We were able to take this new platform, this My Data Helps platform, very quickly spin something up where we can start to capture passive, bring your own device data, BYOD data. So it didn't matter whether you had a Fitbit or an Apple Watch, Withings, Garmin, others. If you were capturing these data at home, you could share it with the study. We would visualize that back for you. And then we started capturing symptom information and COVID test as well. And then optionally collecting EMR data. So how did we do? This was our test. We called up, again, our usual suspects, our wonderful collaborators and partners. These include Walgreens, Fitbit, CVS, but many others. I should mention WebMD, Withings, Garmin, American Osteopathic Foundation, the San Diego Blood Banks. Many of our partners kind of contributed to getting the word out there. We launched March, I can't remember the date, I think it was March 15th, it might have been the 25th. Over the first 4 months that we were live, we enrolled over 35,000 participants all over the country. So you can see it's a little bit small, but the map will show that we had people all the way from Alaska to Hawaii and everywhere in between. So looked at that, we're able to analyze our cohort's data, also contribute to a lot of other collaborators across the world. And we launched our first paper in fewer than 7 months from launch on Nature Medicine with an algorithm that was able to detect COVID or differentiate between COVID positive and COVID negative patients. We have a couple of papers that are coming up shortly on the same data set as well. So, so this is really the way we're trying to build on all of our different domains that Ed's going to get to here in a second. Can we build lightweight foundational protocols built on using previous research to drive new analyses? Can we engage large participant cohorts who are eagerly contributing to the research, getting valuable information back? And then can we take this, can we build out novel algorithms? Can we start to understand physiological responses to infectious disease like COVID, but not only COVID, like other influenza-like illnesses as well? And then can we build on that also sub-studies that go deeper on certain populations? So can we go deep on a long COVID population? Can we go deep on people who are experiencing different types of things or who come from part of our cohort that are in different demographics? So it's been a really great use case. I think it's been an overwhelming success, and it gives us a little bit of excitement as we move into the other domains that we're also going to be focusing our research efforts on in the coming year. So with that, Ed, I believe I will pass it over to you to talk to speak about the other projects that we have going on.
Definitely. Thanks, Katie. And really, it's a beautiful stage setting for what it is we're looking to continue to grow, how we're looking to continue to grow in terms of the Digital Trial Center. As Katie mentioned, Detect not only became a use case, but a success story and something that we can continue to build on, and really is reflective and illustrative of how we can start positioning in terms of looking at other research platforms. And so we really want to move beyond thinking about just deploying studies in a very narrow research context, but move towards more building those foundational elements that allow for progressive efforts to answer research questions in the realm of infectious disease and maternal health and precision nutrition, precision medicine, heart health, and sleep medicine. And this list hopefully will continue to grow in terms of the various different types of platforms that we look to build. And so I'm just going to touch on a few foundational elements that kind of feed into how we go about approaching each of these different efforts. Obviously, it starts with the team. So it's most certainly not just Katie and I. We are backed by an extremely talented team of researchers, clinician scientists that allow for the breadth and depth of study design that we can ultimately deploy in each of these different research contexts. And so building on and pulling from all these different disciplines really allow for that power and strength to be innovative in each of these spaces. So I'm also excited to announce through amazing efforts of the Scripps Research comms team and through Katie really pioneering how we want to describe our efforts in the Digital Trials Center. We went live with the digitaltrials.scripps.edu, so shameless plug for, but also now an inventory of all the different types of work that we're looking to accomplish here at Scripps Research. So I'll just delve into each of those elements that Katie mentioned with respect to Detect. We can now think about them more broadly, more generally in terms of how is it that we can, for example, look at the data with respect to not only the population level, what does looking at personalized information really mean, and what are the tools and resources that we need in order to extract new observations and really contribute a set of relevant findings back to the scientific community. So this includes things now in DETECT. We're obviously looking very heavily at things like physical activity and sleep and heart rate as it relates to a potential predictor of onset of viral illness. We can now potentially apply the same types of strategies, same types of approaches, and in some cases, the same types of modeling in terms of looking at things like blood pressure changes or continuous blood glucose in terms of how it relates to nutritional intake. In addition to that, I think this, for me, is one of the most exciting and important and often overlooked aspects, and that is how can we bring our expertise? How do we continue to build from study to study? How do we truly iterate and continue to innovate in terms of building a cohort, engaging, recruiting, and retaining individuals in the various different efforts that we do? And so this requires knowledge around just even knowing how to define or what a value proposition is. What is it that the participant is actually going to get out of being part of these studies? How do we establish the appropriate incentives, whether they're financial, non-financial, things that are built on and driven by altruism, understanding the different communities that we're working with, what are the motivations that would really drive them to want to be a part of the various different efforts that we have. And as Katie alluded to multiple times now, really tapping into our partner base, really understanding what it means to work with a national partner, what they can bring to the table, what it means to work with local grassroots organizations. And ultimately, all of that is really been combined in each of the different studies that we design and ultimately deploy. Of course, we're very specific in the language that we're using here in terms of study not necessarily being equivalent to platform. Platform, as it is intended, is meant to really convey building of this initial base, and then how do we continue to layer on top of that. So DETECT will not end with its initial conceptualization. We'll continue to build on top of that. And we view the same type of approach for all the different other efforts that we have, whether it means sub-studies that allow for deep phenotyping, or how do we look at sleep now across the different cohorts that we've built. Can we build on specific research efforts that are focused on health outcomes of vulnerable populations? And so this mantra of piloting and then scaling and continuing to explore, I think, has served us well, and is exciting to be able to apply this to all the different research interests that we have here at the center. And then, of course, again, expanding on what Katie described in terms of being able to leverage our technology very specifically, our strong collaboration and partnership with Care Evolution, making use of their My Data Helps platform that allows for a very quick spin up of apps that are participant facing, that really serve as support to the participant in terms of stepping through various different study activities. They have worked with a number of different collaborators. We've already integrated the My Data Helps platform across all of our research studies. And the power here is leveraging things like the commercial off-the-shelf functionalities that it brings, whether it means connecting natively to Fitbit devices or to devices that are compatible with Apple Health Kit or Google Fit, being able to leverage their instance of FHIR-mediated patient transfer of electronic health record data, and just the expandability and customization that we can do within the app to serve our research efforts as needed. So they've been tremendous partners, and we continue to learn and grow together in terms of how to best appropriately use the technologies that they have and how we continue to advance and enhance those efforts. So I'll end with just a quick snapshot of some of the different studies that we have. So kind of hopefully conveyed a bit of our philosophy and some of the infrastructure that supports our efforts. So I'll give a few specifics in terms of how we've implemented all of these strategies. So the first is progress. So this is the prediction of glycemic response study. I actually have a separate talk within the Spectrum series that focuses with a little bit more depth on progress. Exciting for us in terms of really truly applying this remote siteless digital trial model in terms of looking at individual glucose response as it relates to individual food intake. And then excitingly applying this to a population that is diagnosed with type 2 diabetes and comparing that to their normal counterparts. And so this will be going live very soon. We have already a landing page and a press release describing our partnership and collaboration with Tempus, who's sponsoring the study, and ultimately really trying to get at answering two main themes of questions. I think there's some clear biomedical research questions that we could potentially answer with progress, and there's a lot of operational questions that we can answer as well. Can we truly pull this off in a completely siteless digital manner in terms of having participants wear a Fitbit for an intense 10-day tracking period that also includes logging of their food meals, logging of their food, their meal intake, wearing a continuous glucose monitor, and obviously providing biosamples prior to the start of that 10-day tracking period. So a lot being thrown at the participant, a lot for us to learn, a lot hopefully for the participant to also learn about themselves in terms of their health behaviors. Similarly to Detect, that is, Power Mom, born from an opportunity to make use of the challenges that we saw arise during the pandemic. COVID-19 showed a clear, sadly, another example of health disparities, and also showed a deficit in knowledge of how the virus potentially impacts vulnerable populations, and allowed for us to really use that as momentum to revisit a study that we had deployed previously, and that's Power Mom, kind of reskinning this and rebooting this, bringing it onto the My Data Health platform to address a range of knowledge gaps in maternal care, including an opportunity to capture information as it relates to COVID-19 while also being pregnant. So this is being led in terms of a PI for the study by our own Lachey, who is herself just gave birth, a true power mom in all sense of the word, and also a true visionary in terms of how we seek to really change our understanding of maternal health beyond just the episodic clinic visits, really moving to something that's more continuous in terms of our understanding of symptom changes, our understanding of continuous changes that can be recorded through a variety of different digital sensors and devices, and an opportunity really to bring this as Katie mentioned, a foundational element to anyone, anywhere, really broadening that access, especially to those populations that are typically underserved, underrepresented. So an exciting opportunity to really push forth our knowledge and understanding in maternal health. And as always, we lean not only on our team, but our partners as well. Lachey has been working feverishly prior to delivering her beautiful baby daughter in solidifying partnerships not only with United Health Group, but with Cleveland Clinic and St. Mary's Medical Center and Black Mamas Matter Alliance, really to allow for those partnerships that will help us in our mission to ensure that racial and ethnic disparities are something that we can most certainly speak to and capture in this particular effort. We are so excited that we're looking to launch this hopefully in the coming months, even though we don't have dedicated funding streams to this. So a plug for those that are interested in partnering in this effort, because I think this is a perfect example of a platform that has tremendous opportunity in terms of additional sub-studies to follow. And then, so we have Detect that is deployed, Progress that will hopefully be up and running within the next month, Power Mom nipping at its heels in terms of being launched, and then also in design, more in the conceptual phase at this point, is our sleep medicine platform. So Refresh, and in case you haven't noticed, we love our acronyms, and this is our research framework for exploring sleep health. Another exciting opportunity for us to truly build this platform approach, which certainly can have a number of different threads that evolve out of it, including not only just a baseline look at sleep across populations, but now with our technology being able to include things like capture of active task data. We also have a partnership with Philips where we're looking at bringing in some of their sleep devices that really pivot for me in a direction that I hope we go with all of our studies, and that is really starting to dip our toes in the interventional space. How do we not only inform on health behaviors, but potentially provide data that directly allow the participant to change those behaviors as well. So this is just obviously a high level capture of what it is we're trying to achieve here at the center. Extremely excited. We are always actively looking for partners and collaborators across these and potentially other areas that we haven't mentioned or discussed. So please don't hesitate to reach out. And thanks again to Ali and to the organizers for giving Katie and I a stage to describe our new efforts here at the center.
Thanks, Katie and Ed, for the great talk. So I'm going to be talking about, again, digital trials and genetic risk scores, cutting edge technology to transform clinical trials. For those of you who don't know me, I'm Ali Torkamani, Director of Genomics and Genome Informatics at the Scripps Research Translational Institute. I'm going to be discussing sort of a specific use case in terms of polygenic risk score and its application in a platform that we built predating some of these digital trial center efforts, a platform called MyGeneRank, where we are disseminating genetic risk information about coronary artery disease and studying people's response to it. I'll also touch a little bit on some of the other research efforts we have going on, but that'll give a sort of specific dive into a specific use case here. So before I begin my disclosures, I'll just give you a second or two to look that over. A lot of what I'll be talking about and what you heard perhaps in the first session relates to predictive genomics or genotype first health decision making. And what I mean by that is just flipping the way we think about using genetics and sort of control of our health and clinical decision making, as well as lifestyle and sort of behavioral changes. So the way that genetic information is traditionally used is in a phenotype first approach. Typically, you won't get genetic testing done unless you have a rare disease. We previously saw a talk from Phil where he mentioned sort of rare disease cases where these are children that have an overt condition, and they'll go through genome sequencing to try to identify a diagnosis. And that tends to be true for non-rare conditions as well, familial breast cancers, that sort of thing. The testing often is reactive unless you have a very strong family history that indicates you should be tested. Otherwise, the testing is sort of done in a reactive manner. So what we would like to push towards is a genotype first decision making, where you're making a prediction about future health state, and then using that prediction to sort of make either personal health decisions or clinical decisions, etc. And it's not far from what we typically do. Risk prediction is used in prioritization of health decision making all the time. It just doesn't use genetics routinely, and that's where we'd like to get. I talk about this topic in a lot more detail in a prior talk I gave at Future Individualized Medicine 2 years ago. You can see that talk here on the Scripps Research Translational Institute YouTube channel. Cut myself in a flattering pose there, but I go into a lot more depth here, talking about the genotype first approach as a ethical approach to sort of profiling people, and perhaps even in insurance scenarios, that sort of thing. And also talk a lot about just the basics of the concept. But I'd like to review some of the basics. I know there's people in the audience that are maybe not familiar with polygenic risk, so I will review some of those basics before I get into our platform and the results. So basic sources of genetic risk are monogenic risk and polygenic risk. This is definitely an oversimplification. It's really a spectrum in between. But what we're typically used to thinking about is the monogenic risk side of things, meaning monogenic, meaning single gene, essentially. So these are sort of genetic risk that is born out of severe mutations that impact the function of an important gene in a disease process. And that single gene event, sometimes it can be multiple sort of omnigenic or oligogenic, but often a single gene event can lead to very high risk for disease and interventions based on that. So examples in the common disease space include BRCA mutations for breast cancer, which I'm sure plenty of people are aware of, familial hypercholesterolemia and coronary artery disease. But these are extremely rare sort of situations that lead you to high risk for a common disease. So typically, if you test negative for these rare monogenic conditions, it doesn't really tell you a lot about your risk overall. You've only excluded a very small amount of the genetic risk. But although this sort of testing is extremely useful, it is still useful in an unbiased way, and then extremely useful in individuals with pre-existing conditions with family history. Polygenic risk is the other side of the spectrum, where instead of single gene events that increase your risk dramatically, it's just a combination. It's the load in your genome of more common genetic variants that we all share. We all have on average, we're all average in terms of our polygenic risk. These are shared genetic variants. They individually increase your risk by or decrease your risk by very small amounts, and it's the total burden of those genetic variants that determines your risk. And you can put people in high and low risk spectrum based on their polygenic risk. So I've previously reviewed the utility of polygenic risk. Briefly discuss it here. If you want to learn more, I encourage you to look up this review that I've written. But essentially, there are sort of three major areas where polygenic risk estimation could be useful. It's for the prioritization of therapeutic interventions, preventative therapies. Example including lipid lowering therapy to prevent a first heart attack, and that's the example that I'm going to review the use case that we've gone after and the data that I will show. And then there are genetically informed disease screening use cases. So for example, you could accelerate traditional screening, mammography screening, for example, or initiate a non-invasive screening. We heard from Phil about the GRAIL Gallery test. You can imagine if people at elevated polygenic risk might prioritize getting that Gallery test over people with low risk, or people with monogenic risk certainly should consider getting that test. The other side is for screening tests where the evidence is so-so in terms of its net benefit to the population overall. It can improve that risk to benefit ratio. So examples include prostate-specific antigen testing, PSA testing, glaucoma screening. These are screens that are typically not recommended to do routinely, but if you're at high because of false positives and kind of issues with doing the screening in the population, but if you can prioritize people who are more likely to have a sort of positive result, you can shift that risk-benefit balance. And then there's genetically informed life planning as well. I think this is especially pertinent in the neurodegenerative and neuropsychiatric space, where there's interesting work being done on risk scores there as well, showing genetic prediction of educational attainment, for example, relates to it can relate to your progression in neurodegenerative diseases. In neuropsychiatric cases, the polygenic risk scores can sort of predict what components of that neuropsychiatric disease you would have. They can sort of help gauge disease progression or phenotypic sort of advancement of a condition where perhaps there's less to do to intervene. Actionability requires context, and I think that's where these digital tools really come in. They provide the context. So, a genetic risk report, you could just return their risk result. It's not that informative, I would say, to just return a genetic risk result. You really want to return, especially for common late onset sort of complex diseases, you really want a comprehensive view of the person's risk. And that's just because of the way diseases are mediated. Here on the left side, you see for many common diseases, on the left column, we have the genetic sort of contribution to disease risk. On the right side, the environmental contribution to disease risk. And you see it's about 50-50 approximately, depends on the condition, of course, but they both contribute to your risk. So genetics on its own is not enough. The sort of clinical measurements and all on its own may not be enough either. And that context sort of dictates what the downstream actions are. Genetic risk early on might dictate early prevention. Genetic risk learned later on, early prevention in terms of lifestyle or behavioral kind of watchful waiting. Whereas later on, that it might take a more active role in terms of screening or actually taking on preventative treatment. Give you a specific example to sort of highlight this. Polygenic risk for obesity. On the right side here, you see the stratification of a polygenic risk score for obesity, where children, when they're at birth, there's no relationship with their weight to their weight as an adulthood. No relationship between their polygenic risk and their birth weight. But in adulthood, you see this nice stratification of morbidly obese and obese individuals in the high risk group versus, a preponderance of normal BMI individuals in the low risk group. So context is important here because this information is not that useful as an adult. It's useful as a child, but turns out once you're around 8 years old, your current weight is a very good predictor of your future weight. So that context matters, and that sort of determines what the utility is there to maybe lifestyle behavioral interventions early on in childhood. So in this light, we've put together a platform for decentralized real-world genetics research. This real-world sort of topic has come up multiple times today already, where I think the importance is how does this stuff operate in the real world, and can we generate evidence for the utility of these platforms using real-world genetic data at scales where we can start seeing interesting signals. So we built this platform to essentially empower research participants with contextualized genetic risk, meaning that it's a platform where they can receive their genetic risk, but it's got integrations with other health factors so that we can contextualize that risk as well. What's shown on this slide in black are capabilities that existed when we first sort of launched MyGeneRank, which the results I'll show you relate to. In gray are capabilities that we're still in the process of building. And I'll just highlight that we're able to bring in genetic data automatically from 23andMe and working on a connection for sort of de novo generation of genetic data using the Quest patient service centers. On the health data side, you have mHealth integration, of course, and then we're working on EHR data integration, so you give more objective information about health status and outcomes. Ultimately, the way we see this is a platform where the participant drives the interaction with their risk. They're educated by essentially the interface, and we hope that sort of promotes self-efficacy in preventative behaviors. And then there's the flip side, the provider education side, where the goal there is to sort of give them confidence about the use of this information and understanding about the utility, so they can sort of mediate that shared decision-making process. So I'm going to jump into the use case, the specific use case now that we initially launched MyGeneRank for, and that's polygenic risk in coronary artery disease. So I'll review briefly the utility scenario. So for coronary artery disease, or heart attack, essentially, when you go to a physician, they essentially determine your 10-year risk of having a heart attack, and based upon that value, at least based on clinical guidelines, that sort of dictates whether or not you need to be on lipid-lowering therapy or not, or a statin. These are on the left side here, you have the different risk tiers and the guidance that's given. So essentially, if you're intermediate risk in the 7.5% or greater risk group, it's suggested that you take a moderate intensity statin. And if you're high risk, you should be on a high intensity statin. For primary prevention, being the prevention of a first heart attack, if you just follow these guidelines without any genetic information, you have a number needed to treat that is relatively large, and you need to treat a large number of people to prevent a single heart attack, and adherence to these guidelines is quite low, somewhere around 30% adherence to the guidelines. Polygenic risk allows you to identify not only people who are at higher risk, but in the coronary artery disease case, identify people who are at higher risk and who receive a greater benefit. So in the high risk, high polygenic risk group, there's about a 20-to-1 sort of individuals treated versus heart attacks prevented. Average genetic risk sort of has an average result. And on the low risk side, potentially there is not much benefit from going on a statin. So I'll refer back to this. Low genetic risk is individuals who have a coronary artery disease polygenic risk score at the 20th percentile or lower. High genetic risk, 20th percentile or higher, and then average is the 60% in the middle. There's a lot of different ways to define these risk tiers, or you can do it continuously, but these are the risk tiers that we define up front. So here's the MyGeneRank platform, first-in-class app for returning this risk information. What I've highlighted here in red is the platform that was sort of in use as the results were generated, where most people actually worked on this very baseline platform on the left side. And then we've had this progression improving the platform overall, which can talk about later. But so we have a basic platform with a risk reduction interface. It shows you your genetic risk overall, as well as your genetic risk in context of these this clinical risk that I mentioned. You can modify your behaviors, active lifestyle, healthy diet, healthy weight, and see how that changes your risk. In later versions that haven't been used in this study so far, will allow people to actually turn on and off a statin or change their lipid levels, etc., to see how that changes your risk. And we've also done a lot of work in the background, mechanical Turk experiments, etc., to improve the interpretability and understanding of that user interface. The other thing I would say is that Polygenic, we also updated the score in the middle of this study, and we learned a lot from that. The score improved. We felt that we need to return an improved score. There had been a lot of advancement in terms of the understanding of coronary artery disease genetics, and that leads, of course, to some people's scores changing. So there's some noise in these results. Another interesting finding is that the tools that you typically use in population genetics don't work. They have some issues when you apply it to individual level return of results. And one of those issues can be variability in the result when you use population genetics tools from run to run. So here's the participant population. We had about 3,800 individuals at baseline. 243 individuals responded via the app without any prompting. 641 people responded after we sort of pestered them with emails. Here's and a little bit later, so about 1 year follow-up for the proactive responders, about 1.5 year follow-up for the reactive, longer-term responders. You see here the risk distribution. Actually, most of our population is low risk and doesn't necessarily need to be on a statin. But the respondents here, if we look at the subset of people where we could determine a CVD risk score, we have an enrichment of the intermediate risk people where there's the most utility responding to us in terms of what changes they made. Here's the ancestry and ethnicity distribution. We could definitely do better on diversity here. We do have a Spanish version of the app as well, but clearly we need more effort there. And here's the age distribution as well. Again, older, perhaps higher risk people enriched and responding, and about 50-50 males, females. 16% of the baseline participants were on a statin. About 27% of the respondents, people that responded, were at baseline on a statin. Here's their reactions to the information. So when we return the genetic risk information, what do they think about it? My genetics make it more likely that I'll develop coronary artery disease. You see a nice sort of agreement. This is a Likert scale where 5 is strongly agree and 1 is strongly disagree. You see people that have higher genetic risk report that they agree with the statement that the genetics put them at higher risk. So we have good understanding on the genetic risk side, not so great understanding actually on the clinical risk side, on the baseline application, which is where we've made some improvements. Anxiety and worry is a topic that comes up often. We see very little evidence for that. Slightly higher worry in the high risk group, but a response of 3 is actually neutral, and perhaps they should be a little bit worried. And then self-efficacy question, I'm able to reduce my risk, is about even. Everyone believes that they can reduce their risk. All right, now intentions. So once we return results, we ask them, what do they intend to do with this information? through this question that's listed here. And about 20% of people in the high-risk group say they intend to increase their use of a statin, and that sort of correlates with risk overall. And then you see that also on the decrease use side of things, a higher percentage of people on the low-risk side saying that they intend to discontinue the use of a statin. And most of these people reporting in the decreased use side are obviously already taking a statin. You'll see, it turns out that the discontinuation really doesn't happen much. Here's the actions in the short term. That is those people who responded via the app proactively. We asked them whether or not they made any changes to their use of the statins. And again, you see a nice sort of correlation with genetic risk, where in the high risk group, you have a substantially larger percentage of individuals reporting that they increased their use of statins, as well as other lipid lowering therapy. You have a nice signal here of individuals initiating both statins as well as other lipid lowering therapy. The picture changes a little when you look at the longer term follow-up, the reactive respondents, perhaps a little bit less engaged. You have an enrichment in terms of this is all initiation of any lipid lowering drug. You have an enrichment again on the high risk side, but not quite as strong of that kind of correlation with genetic risk. And ultimately, this is what the picture ends up looking like, where at follow-up, you have nice stratification of individuals, especially for total, the rate of individuals using lipid-lowering therapy in total, where you have a nice stratification of low risk. The low-risk side doesn't see a dramatic increase in use when you have this sort of graded effect across genetic risk, kind of showing that the genetic risk is sort of guiding or aligning preventative therapy with a level of genetic risk overall. So we have this, decrease in the signal on the long term. How can we improve that? As I mentioned, we've made a number of changes to the platform since then. On the left side here, you see a new sort of risk reducer interface that we've developed through a bunch of crowdsource-based testing of interpretability and understanding of this interface, improving the risk reduction interface, giving participants more control over what factors they can adjust to see how their risk changes. And then all these additional key capabilities to build that ecosystem to provide context and support to reinforce that information. So in the last couple minutes, I'm going to talk about how we see this moving ahead. So what we are about to launch is a more formal prospective study looking at the clinical utility of polygenic risk estimation, incorporating some of these more objective health measures like EHR measures instead of the self-reported survey-based approach that we've taken. And the goal is really to prove clinical utility. So I would say the clinical validity is there. These risk scores do stratify people by risk. Perhaps there's even the therapeutic benefit in the high-risk individuals, but we still need to prove the clinical utility in the real world. When you distribute this information in the real world, does it actually make any difference to health outcomes in the long run? Is what we're really interested in using PEP versus prospective electronic polygenic risk study. Another area where we'd like to explore is polygenic risk use in molecular autopsy. So I'm not touching on the wide variety of other sort of genomics projects that I lead, but one of them is molecular autopsy, where we're doing genome sequencing in individuals who have died suddenly and unexpectedly in San Diego. Recently did a reanalysis here and identified a couple of more causes of sudden death and other rare conditions. You can imagine that early sudden cardiac death is a big problem. It's one of the leading causes of death in the developed world. Some of this is likely mediated by polygenic risk. You could imagine also if someone dies early from a heart attack, let's say in their 50s, besides familial hypercholesterolemia testing, especially if there's no sign of very out of whack cholesterol, polygenic risk may be sort of the cause there. And you can imagine doing polygenic risk cascade testing just as you would do monogenic cascade testing in at-risk individuals. It's really important to develop these platforms and resources locally, and that is due to population stratification. So the way that these scores are generated is by very, very large genome-wide association studies where you have now hundreds of thousands of people being genotyped. The UK is a big leader in this. Turns out that your genetics, there's population level stratification as well, and it's not just white versus black or Asian individuals. There's stratification even within European individuals, and that genetic stratification can lead to score biases, especially if the genetic stratification is related to other confounders that might increase risk, that would sort of lead to you thinking that some genetic variants are associated when they're not. So local resources, I think, are really important to making sure these scores are applicable to the environment that you're rolling them out in. So, another reason to really develop both genomic resources as well as these platforms here. We're pursuing integration with deep learning as well, where we're trying to improve these scores. So these scores are relatively basic, I would say. You determine a clinical risk score, a genetic risk score, and those are essentially multiplied together. It's not true integration of genetic risk factors with other risk factors. So we're attempting to do that using a deep learning approach. And the key here, I think, is these interactions between genetic risk and other factors is key to the individualized medicine component of this. So the statin effect is an interaction, essentially, of your risk with lipid-lowering therapy, which drives the decision of taking on a lipid-lowering therapy. Those sorts of interactions, I'm sure, exist in a number of other areas, blood pressure, other intermediate risk factors that relate to coronary artery disease. So if we can dissect those further, then I think we can improve the actionability further. And I will end on this slide. I think it's somewhat obvious maybe that we want to head towards a dynamic, context-aware risk estimation and health recommendations, where you have this app-based platform that you can use to collect and return health information as well as genetic risk information. It makes the research a bi-directional process where the researcher and the participants are both benefiting from this interaction. Of course, as you generate more data, then we can improve these deep learning models and improve the genetic risk estimation, as well as the recommendations that you could make based upon those recommendations. So connecting these resources together. And finally, this work is done by a very large number of people besides myself, the Scripps team on the left side, as well as our interns on the right side. And with that, happy to take questions. I will start with one for you, Katie and Ed, on the COVID-19 infections. What learnings do you think are transferable to clinical trials outside of infectious disease, where patient compliance is perhaps not as high? And do you think digital tools could improve compliance?
Yeah, I definitely think so. I think as we move toward an environment where we're making it much easier for people to participate in trials, and we're remembering to give that value at every stage, I think we'll see an improvement in compliance for sure. I think, but that's the critical piece. So as you move away from that traditional sort of at the site, trusted physician recruiting you in person, where there's a lot of just natural give and take and trust that we sometimes take for granted in our face-to-face world, maybe not as much anymore. But as we move that into the digital space, we just can't assume that stuff is still there. You have to build it into the experience. And I think as we get better at that, we'll see improved compliance because it'll just be easier, and we'll be able to integrate research into people's lives.
Yeah, I agree. I think the in-person sort of experience can perhaps lead to, or easier, I think, to initiate that engagement in person. But and we've seen in many other studies that long-term engagement, I think, is difficult either way, digital or in person. But yeah, as long as you can keep a bidirectional, I think the bidirectionality is key there, right? To keeping people.
Sure, you've got to add value at every stage for everybody on both sides of the relationship. So.
All right, we have a question about the sleep sensors. Do we know how accurate and reliable they are at capturing true sleep stages and sleep cycle?
Yeah, so I can address that and actually broaden it out a little bit because it's a question that comes up quite a bit, but specific to sleep. So we have an opportunity to leverage wearable devices like Fitbit and other physical activity trackers and smartwatches that collect different levels and robustness of sleep data. And that's true for physical activity, it's true for heart rate or respiratory rate, skin temperature. So it's important to recognize that what the context is in the collection of those data, but also what it is you're trying to model. And so for something like Detect, where we're looking at variation from baseline, that's very different than comparing steps from person to person. For example, you truly are looking at, all right, what is now deviating from an established baseline that can speak to a potential difference. And so we see that with Detect across those three, actually, sleep, physical activity, and heart rate. But circling back specific to sleep, what's interesting about the partnership with Philips that we're still working on, as you know, Ali, is actually now bringing in a device that is introducing a potential intervention. So a little bit of a different twist there in terms of not necessarily specifically collecting sleep data, but now seeing what an intervention could have in terms of impact on that. I'll just quickly end by saying and give a plug to the Digital Medicine Society, the DIME Society, that has launched the playbook. Highly recommend for folks to check that out. It is an extremely in-depth look and survey of precisely this question. What exactly is a digital measure? What is the difference between a device and a sensor? How do you integrate it into clinical studies, validation considerations, and so on?
Yeah, I think the pre-post sort of aspect is critical, right? It's sort of self-calibrating, I guess. I'll just comment from personal experience. I think perhaps the sleep phase, not sure about how well that's determined, but other measures like heart rate, etc., that are sort of objective single measures, determining that overnight while you sleep, I think is also really interesting. So there's a lot of information that goes into that sleep phase that I think is really untapped sort of continuous information for improving these more synthetic phenotypes like sleep phase further. All right. Could you speak a bit more about the process of returning any potential actionable results to participants? And there's a related question. about for me about incorporating whole exome sequencing and whole genome sequencing data in these results rather than GWAS SNPs. So the data that we get from 23andMe doesn't have these, the monogenic component, essentially, familial hypercholesterolemia, et cetera. So we don't return that information. We haven't quite built the capability for that yet, but we are working towards it, with the Quest sort of platform where I mentioned, where we have a little bit more flexibility with the sequencing platform, where we can layer on top these monogenic components as well. And the process of returning these actionable results, at least for the polygenic risk side, it's entirely app mediated. And automated. Genetic information comes in, goes to our systems, gets calculated, the result comes back to you, and your survey answers essentially populate the clinical risk component of it. On the rare disease side of things, we have a very different process that involves a physician champion, personal communication with the individual. These are individual results of very high impact, in which case we take a very different approach to the return of results.
Yeah, and I'll say for the larger programs that we run, including the All of Us research program, the general idea is that any information you share with us, in the Digital Clinical Trial Center is your information, right? But you should also get a choice on whether or not you receive that information back. We're not going to give you that information if you don't know it, but if you want to know any information about the data that we collect on you, it is yours to know and have. For something like actionable results for the All of Us research program, what we've done is work with them to set up an infrastructure of genetic counselors and physicians that can explain things to you, and then we're working very hard to explain the results back to individuals as well. All
right. There's another comment on that. I think the opt-in kind of opt-out is the way to go, right? Just in general, I would say genetic risk-wise or otherwise, even including non-actionable results, I would say, but not everyone agrees with that viewpoint. All right, we are out of time. I think I will end there. There's a number of other questions about polygenic risk and obesity. I'd be happy to address those in the networking session that we're about to go to in 2 minutes. So thanks, Katie and Ed, for the great talk. Thanks for everybody. Thanks everyone for attending. I would encourage you, Ed has a focus talk that he's giving in the R&D sort of section as well. So check that out. Go back to the website, to the R&D tab. You can see Ed, as well as a number of other speakers talking about some of the studies that were mentioned, Power Mom, et cetera, in greater detail. And other than that, please navigate back to your agenda page and join a networking session. We've opened them up to everyone. Thanks a lot. I'll see some of you there.